A wave of new technologies is sweeping across biology. Flow cytometry can now read dozens of markers simultaneously from hundreds of thousands of cells every few minutes. Functional MRI can deliver a movie of the brain at work. We can monitor the activity levels of tens of thousands of genes and score millions of DNA variation sites in an individual. For less than $10,000, we can sequence entire human genomes, and $100 genomes are already in sight. Plummeting costs mean that within a year, individual labs all around the world will be gathering terabytes or even petabytes of data. These data are already being linked to an array of other information to offer a fundamental new understanding of living systems. We have crossed a threshold into the realm of the new biology.
I don't know whether the correct term is the new biology or the golden age of biology. I just know that the level of enthusiasm that I see right now in the community and the way it is coming together through large integrative processes is unprecedented.
We are at the beginning of a new era. We call it a new paradigm.
The new biology, as it's evolving today, is basically many different fields realizing that they can collect a lot of information. They can bring to bear computational tools to mine the data that can give us results that are correlating to both biological and clinical outcomes. The goal of any kind of research is not just to create information, but to actually distill it to some level of wisdom. That's the goal. Data, information, knowledge, wisdom.
I think the new developments in ultra-fast sequencing will take us into a new era again in biology.
The new biology will transform every aspect of human health, from food production to cancer treatment to disease surveillance. And this scientific revolution has one organizing theme: assembling complex, high-dimensional bits of data into a realistic portrait of life.
There's a big realization now that actually we have to put it all back together again to really understand the intricacies of the very complex systems that we're looking at. That's been really difficult to do, but now with new technologies, new understanding, which is very much founded on our previous understanding, we're beginning to be able to do that. So it's an incredibly exciting time.
The new biology is systems biology realized. And it's realized by the new technologies that enable us to take the comprehensive snapshots of the system, but it's also enabled by the new ways of thinking, of integrating information, building complex models. The human mind is an amazing engine for putting information together and understanding it, but it has to be presented with the information in the right kind of way.
Think about how your mind organizes data, sound, pictures, and text, to let you grasp a story and feel its emotional impact as you watch this Google ad.
Each movie is a frame of millions of pixels, and then you have thousands of frames through the course of a movie.
If you looked at the ad the way that old biology looked at living systems, you'd see each pixel as an average value over time.
Say, okay, understand this movie from this average of all the pixels. You'd never achieve understanding that way.
Or you'd look at one raster line and see how the pixels' intensities change over time. It's more information, but meaning is still far away. Now, add a bit more data, fragments of frames, a word here, a symbol there. Next, add context. The words are appearing on a screen. Whose computer is it? More context. Someone is performing a series of searches, asking questions. Now you see a bit more, but not in the right order, and you can't tell what's most important. Ideas are flying in your mind, but they don't add up to the story the ad intended to tell. Now add information about which words are most important, the ones in the query box, but still you lack the right order to get the truth of the story. You might come up with something that seems sensible, but is wrong. Someone is planning a vacation in France or looking for a job with an airline. If you add sequence information, you'd get a partially true story. But with information gaps, you can miss the whole point of the ad. It's a love story. Now, bring in a new layer of information, sound. And at last, the entire true story is revealed with full emotional impact that completes our understanding. Data, information, knowledge, and knowledge about that knowledge. That's what you need to achieve wisdom. Can we look at living systems in the same big picture way we watch a movie? The old way of doing biology was a lot like looking at a few pixels averaged over time. You'd study a handful of proteins, and on a good day, you'd end up with a simple linear pathway that explained a few of the steps that cause disease phenotypes.
There is nothing in biology so complicated that if viewed in the right way does not become still more complicated. We know a tiny fraction of 1% of all there is to know.
I think the real promise of molecular biology was, for the first time ever, we could look at the individual components, that is, the individual genes that encoded the information for making organisms. And I think in a naive reductionistic view, people thought by understanding the individual components, they could come to understand the collective behavior of those components. And that turned out to not be true at all.
The new biology is corralling data into meaning. The data are large numbers of observations of molecules over time and individuals, and at a hierarchy of levels. Sophisticated algorithms can reconstruct causal molecular networks that represent the physiological states associated with complex traits like disease. These networks aren't islands. They are highly interconnected in a complex system. The intricate multi-scale network of life can be visualized in this model. Each network, defining physiological states of the system, maps to a region. When one network affects another, that's shown by a directed arc. Interacting networks are often found within a single organ or tissue in the body. What happens when you look at the interactions of networks across tissues? These are shown by links inside the model. It's only become clear in the last few years that the interactions across tissues may far outnumber those within. This is a compact representation of a system-wide molecular network that underlies the physiological states of an organism. It enables higher-order predictions of how an internal or environmental perturbation will ripple through a living system. This link between molecular biology and physiology is the central insight of the new biology. What's making the new biology possible are novel bioimaging technologies. Functional MRI, for example, shows brain activity over time. It's become a key tool of cognitive science.
At MIT, one of the cool things that we've discovered using functional MRI is that there are at least a few distinct parts of the brain that conduct remarkably specific functions. One of the discoveries is that there's a region in the right hemisphere that's specialized not just for simple perceptual operations, but this one is specialized for thinking about what other people are thinking.
The fMRI system delivers brain data in the form of activity levels over time of each of tens of thousands of voxels, or 3D pixels. A voxel usually represents around 1 million neurons.
In a typical MRI scan, we scan subjects in there for about 2 hours. We collect about 3 gigabytes of data. My lab collects about 10 terabytes of data a year.
At the level of individual cells, another high-throughput imaging technology is flow cytometry. It can now deliver a gigabyte per minute of high-dimensional data. And DNA sequencing technology has come a long way since its birth.
We've gone from the early machine we developed in the mid-'80s to machines now that are 20,000 times more rapid, less expensive.
Second-generation machines have high throughput, enabling broad-scale human sequencing for the first time. And third-generation technologies are on the way.
The single-molecule real-time paradigm allows you to see the individual components, the characters, if you will, in the play of life, in their appropriate context, but one at a time.
This smart approach eavesdrops on nature's own gene readers, polymerase enzymes, to get DNA and RNA sequences. The same approach can also examine proteins.
In the near future, we will have a technology which could sequence the human genome in $100. But when you have these $100 genomes, how you could read it, how you could understand it, how you can use it.
One effort to exploit massive amounts of genetic data is taking place at the University of Tokyo. Biobank Japan has data representing the DNA of 300,000 patients spanning nearly 50 diseases. Yusakai Nakamura is the founder of the project.
それらの病気の方の DNA あるいは遺伝子多形を比較することによって病気になりやすさを見つけより良い治療法を患者さんに提供しようとしています。医療の体系そのものがこのバイオバンクジャパンを通した膨大なその臨床情報からゲノムの情報まで合わせた解析によって変わってくるというふうに考えています。
One more technological advance is needed if the new torrent of data is to be harnessed. A leap in computing power.
We're generating more data than you could ever go back into the lab and validate.
There's an intangible cost with the difficulty in managing this glut of data. It's a logistical nightmare.
その理由は、そのデータの取得が自動化されてきたことなんですね。だから大量のデータが出たと。そうすると大量のデータは、自分の鉛筆と紙だけでは計算できませんから。
We used to think terabytes of data was big, but that's nothing these days. Now we're into peta or even exabyte scales of data.
We now have seas of data which are drowning us. People now are coming together, realizing that the key to mining data is computation. Look at Google, look at Amazon. These are key industries that have realized that there's an awful lot of data that can be measured from the herd, from crowd effects.
Offering deep and novel understanding of living systems, the new biology is already revolutionizing healthcare. And that includes the costliest group of illnesses, cancer.
To be diagnosed with cancer is difficult. To be diagnosed with multiple myeloma, an orphan cancer that's uniformly fatal, is beyond challenging. And then to find out that you have one of the highest risk forms of myeloma is devastating.
Kathy Giusti was a 37-year-old pharmaceutical company executive when she was told she had only a few years to live.
My daughter was only 1. My greatest hope was that maybe I would see her attend kindergarten.
It's not necessarily the thing you want to hear. A very harried doctor tells you you've got kidney cancer. It doesn't look like it's spread very far yet, but I'm going to call urology. I'm sorry, I have to go.
Stanford University immunologist Gary Nolan had heard this kind of news before.
I've had 5 incidences of distinct and unique cancers.
My twin sister has a very healthy immune system, so after receiving high-dose chemotherapy, I ended up doing a stem cell transplant with her. Should I lose my fight with the disease, she's right here to help with my family.
I just got a CT scan yesterday. The radiologist said there was nothing, no evidence anywhere that it had spread. It is taking care of things early, quick action, which is, I think, what some of the network biology that we're going to be able to do in the near future is going to enable.
The field of cancer research has had a long history of taking a very reductionist view of the problem, thinking about just the cancer cell, not the genetic networks that surround the offending oncogene.
In sickness and in health, genes, proteins, and metabolites are highly interconnected, like the strands of a spider web.
There's no place on a spider web that you can pull an element of the web and it isn't felt somewhere else.
Nolan is teasing out the networks underlying cancers of the immune system.
We're not just looking at T cells in isolation and what they are capable of doing, but saying when T cells do this, at a distance, B cells, macrophages, dendritic cells are compensating like a great machine.
Treatments like Excel, right?
We now are using a variety of techniques to determine the different kinds of networks that exist within each cell and then within the interlocking system of the cells as they talk to each other.
Nolan uses flow cytometry, a technique for rapidly examining cells one by one. By increasing the number of molecules he can detect, Nolan offers an exponential explosion in the number of cell states that he can measure.
We can measure 35, 50, soon 100 parameters on a per cell basis. It lets us look at heterogeneity at a more granular level. It lets us describe all the multiple cell subpopulations and dynamic states that are occurring at any given point in time. It gives us quantitative information that allows us to bring powerful statistics to bear. I can ask millions of questions at a time, not just one question at a time.
Because Nolan can detect exponentially more cell types, the number of molecular networks that he can consider shoots up super exponentially. In this new technology, Nolan tags an antibody with one of a large number of isotopes. Each antibody attaches to a unique protein on or in a cell. A cell is shot into a flame hotter than the surface of the sun. The cell becomes a plasma. As the spectrometer identifies the ions, Nolan gets a gigabyte of raw data per minute, which tell him which of dozens of proteins are in each cell, uniquely identifying different cell types. Nolan applied this new technology to acute myeloid leukemia, or AML, a cancer of one line of blood cells. He zeroed in on one lineage of blood stem cells.
These would be cells that represent the tumor as they come right out of the blood of the patient.
Nolan now activated these cells with a molecule that triggers a strong immune response, the context of most cancers. Now, when he ran the cells through the mass spectrometer, he discovered that what had looked like one group of cell types was actually two. The green cells are far more numerous in early stages of AML and can be killed by chemotherapy. The blue cells are another story. The grandparents of a blood cell lineage, the blue cells resist chemotherapy and come back to kill the patient.
We now know that every patient comes in the door with two cancers and therefore should be getting two therapies.
Never before have distinct stages of a cancer been found that show up at the beginning and end of differentiation. Mutations in a grandparent cell derail its journey down the family tree.
The signaling networks within them that are supposed to go from one cell state, they go through a rapid set of changes and then they reassemble like an origami. If you fold them a different way, they become something different.
While Nolan is working at the level of cells, another investigator wants to understand cancer at the level of networks of molecules. Andrea Califano is trying to represent, in predictive network models, the millions of interactions among proteins, DNA, and other molecules. The challenge is that by the time cancer is detected, a cascade of molecular changes has swept through the body.
The most important thing in a biological system is who regulates, who is totally upstream of that process. The single pebble that's sliding down the mountain causes the avalanche. The view of biology that we've had for many years is of observing the result of the avalanche. We use an approach that is really fundamentally based on our ability to characterize the targets of major regulators in bulk. Instead of saying these are the 10 targets of that transcription factor, we have to say these are the 1,000 targets of that transcription factor. This has never been possible before.
In Califano's models, nodes represent gene expression traits, and links between them represent regulatory relationships. Califano can use this map to narrow his search for the causes of disease.
Right now, using these models, we are really able to pinpoint within a very small number of candidates, in the order of less than 10, what was the gene that you had actually perturbed. It requires tremendous computer power. But at the same time, it allows us to go back to these master regulators of disease.
Califano's model also lets him predict the efficacy of drugs.
We bring those predictions to the lab in a highly roboticized environment where literally hundreds of thousands of experiments can be performed in a week. And these experiments can involve silencing genes or gene combination.
Califano's lab can help identify drugs that precisely target the dysregulated networks in cancer, and that's a big step beyond current treatments.
The majority of tumor treatments today are basically just cytotoxic. You're just killing the cell indiscriminately. To a large extent, it's like shooting with a hand grenade into the body.
Any individual cancer treatment will work in anywhere from 50% of the patients down to as few as 5%. We've actually cured cancer. We just don't know who to give the drugs to.
At the Broad Institute at MIT and Harvard, one researcher is trying to help get the right drug to the right cancer patient. Todd Golub doses cells with an array of pharmaceuticals and other small molecules. He then projects this data into existing network models. For each of the 20,000 or so human genes, Golub's connectivity map shows whether expression goes up or down after exposure to each of over 7,000 drugs or small molecules. Golub is exploring the interaction of macrophages, white blood cells, and cancer cells in a tumor.
It turns out that there are different types of macrophages, some that promote the growth of tumor cells and some that actually repress the growth. For the first time, we're going to look at what this signature looks like in the connectivity map database. I haven't looked at this before. I have absolutely no idea what's going to happen or whether it's going to be useful, but we'll see. And we're going to execute the query.
Golub is asking the connectivity map to look for drugs that will change the gene expression of the bad macrophages into that of the good ones.
There it is, so it's done. So here we actually have quite a very strong result where we have a very strong connectivity with a number of drugs. And one striking thing that we can see just looking at this is that there are a number of histone deacetylase inhibitors on this list. For example, vorinostat was recently approved by the FDA. As far as I know, a role in the differentiation of macrophages hasn't been suspected. I have no idea whether this is going to actually work or not, but it suggests an experiment that we never could have thought of before.
Another of Golub's projects is a study of the complete genome of a cancer of blood plasma cells, multiple myeloma. This is Kathy Giusti's illness. Giusti, with her twin sister, started the Multiple Myeloma Research Foundation, which has collaborated with Golub on his research on the topic.
The early returns on this myeloma genome project suggest that there are whole aspects of biology that are entirely new, not only to multiple myeloma, but to cancer biology in general.
And the more we understand the multitude of pathways affected, the more we will find the right drugs and combinations of drugs to cure this disease. I had a certain translocation that made me high risk. It was hard for me to live with that for a while because there was no drug to help me. But today, because we know that that translocation is found in about 15% of myeloma patients, the drug companies have started developing drugs, antibodies in that field.
Giusti's foundation has helped get 4 new drugs approved for multiple myeloma. With these treatments, a patient's life expectancy from diagnosis has doubled to 7 years. Giusti herself is an outlier. She's already survived 14 years.
Now I live year by year. I never believe I'll have more than a year, but I live in that year, and I am grateful for every single milestone I see. Whether it's having seen my children's first steps to their first touchdown, their first home run, their first homecoming game. Where we are now is looking at the next generation of drugs, which we have many in our pipeline. But again, I still believe that cures will come from combining those drugs with the genomic information and deciding who can be cured and with what.
Cancer is a major cause of death worldwide, but infectious diseases kill 20% more people. And the threat of a new outbreak, perhaps the next plague, is only rising.
The level of interconnectivity, whether it be the movement of animals or movement of humans by planes, from the perspective of a virus, we're one single population. We live at this moment in history where we're going to have more and more pandemics. It's just the nature of the way that our species is.
When the Sars epidemic of 2003 jumped from China to Canada, public health experts were blindsided and worried.
If it hadn't been Toronto, it could have been anywhere. And I think that's the ultimate fear we look at with pandemics.
The Chinese government responded to the outbreak by beefing up their command post for epidemic surveillance at their Center for Disease Control.
What you see is truly state-of-the-art ability for a large country, 1.3 billion people dispersed over a huge geographic area, to collect real-time data on over 30 infectious diseases. The Chinese track that data, aggregate it, report it, and then deal with it at a political level in any way that's necessary for the Chinese government to respond to an infectious disease outbreak.
China's disease surveillance system is arguably the best in the world. Around the clock, thousands of hospitals and clinics report large amounts of patient information, but the approach is low-tech, diagnoses and symptoms. The new biology can take disease surveillance to the next level by focusing on places most likely to become the source of a new pandemic, the hot zones of the world.
We do know certain things about pandemics and how they're born. We know that certain host species out there are more important than others with regards to seeding us with new agents. Bats, non-human primates. We know that there are certain sorts of behaviors which are very, very important for cross-species transmission. Hunters, people who have contact with a vast range of wild animals and who have contact with all the various viruses that exist within these animals. And all of those represent opportunities for the transmission of these viruses into humans. But most of the viruses out there that have the potential to spread in humans we're not aware of.
Working in hotspots, virologist Nathan Wolfe takes blood from humans and prey animals. But these are only partially sequenced, and that's back in the US, after a significant delay.
The idea that you could take a drop of blood and find out all of the genetic information in that drop of blood, get a sense of all of the organisms, all of the viruses, all of the microbes that exist within that drop of blood, that's something which we have really not attained, but which would be incredibly important for our field. And the moment that we get it, it'll change the way that we do this kind of work.
Until all potential pandemics can be caught in the first sick patient, the next most important use of the new biology is to slow the spread of an outbreak. and that means early detection of a pathogen's spread. In a pilot project in a large American city, raw data is coming from raw sewage. Researchers collect regular samples from a number of plants. Contamination from sewage, it turns out, is responsible for many outbreaks, including SARS. Samples can now be directly sequenced to detect known and unknown pathogens.
Can we real-time monitor those environments for these different things that make you sick? Things like single-molecule real-time sequencing, able to get whole genomes of pathogens sequenced from collection of the sample to sequencing of the sample to getting the data. Well, what's in those samples? Do I see influenza? Do I see noroviruses or other more severe viruses that would cause public health concern?
Now imagine using sequencing data with fine-grained geography and displaying the results on a new kind of map, a disease weather map. You might see a flu outbreak starting near an airport. You could zoom in on your neighborhood restaurants to see where you shouldn't go to dinner tonight. You also might even get a forecast. Cool colors signify a low rate of infection. Warmer colors show harder hit areas. And people who drive can spread the outbreak like a wildfire. If it's H1N1, you could have Tamiflu shipped in advance, or travel could be limited to slow the spread. Actionable results will be the hallmark of the new biology. Just as the new biology can raise the effectiveness of disease surveillance, it can also transform another area central to human health: food production. The problem couldn't be more pressing.
The big picture is this: 1 in 7 on the planet are undernourished. 13% of the world's population, around 1 billion people, do not get enough food, particularly enough calories. Today, the world's population is very rapidly approaching 6.9 billion. In 40 years' time, in 2050, about 9 to 9.2 billion. How do we feed the people here today that aren't getting enough to eat? And how do we prepare to feed people in the future?
In half a century, we will need 100% more food than we produce today. Added farmland will make up 20%. Another 10% will come from techniques such as planting more times every year. So that leaves 70% of the world's coming appetite that can only be met with technologies that will be led by the new biology. The need to get more out of every acre is an especially acute problem for the world's most populous country, China. This nation has only 7% of the world's arable land, but 23% of its people.
The land is so limited. Those people need to be fed. So we have to use genomic technology to improve that.
China is the world's largest rice producer, and that's due in part to strains of rice called superhybrid, breeds that produce at least 20% more grain than the strains they replaced. In 2004, superhybrid rice was the subject of the World Food Prize. Now, in the port city of Shenzhen, China, an institute called BGI is a key player in a project to discover what puts the super in superhybrid rice. And that means discovering the complex gene networks that control yield.
So we are in the process of deciphering genes that actually make this hybrid rice produce more.
With prodigious technological power, researchers in China have sequenced the 40,000 genes of the maternal and paternal strains that came together in superhybrid rice. But that's only the first step.
The project is not a simple sequencing project, so the biological contents has been expanding to reveal all the genes and to build network. All the genes are regulated at different level. Some at the structure level, some at the gene organizational level, some of them at the microRNA level, and their mechanism at the protein level.
Understanding the complexity of yield under different conditions, for example, flood or drought, demands the new biology. By generating and integrating large-scale DNA variation, molecular phenotyping, and trait data in plants derived from superhybrid rice strains, we can reverse engineer the molecular networks that control yield. We can then target the best subnetworks to crank up yield under any environmental condition. And environmental conditions are only getting more challenging given both climate change and the bringing of marginal land under cultivation. Information about molecular networks is already speeding the development of new strains of another staple crop, corn.
People have been breeding corn and other crops for thousands of years. Since the last 50 or so years, the rate of increase in yield really has not changed that dramatically. And so in order to feed the world in the future, we really need to rethink how we do breeding and how we produce agricultural crops. How are the yields looking?
I think they'll be really good this year. We've had plenty of moisture, so we should have good grain fill and good yield.
Yeah, it looks like it's filling in nicely. Breeders can send us several ears of corn, and we can tell them which kernels from those ears are going to have the highest yield potential.
Growers from all over the world ship corn to Monsanto's high-throughput genotyping center in Ankeny, Iowa. Each kernel in an ear of corn has a random mix of the parent's genes. Instead of growing every kernel to see how the plant turns out, Monsanto is using the techniques of the new biology to pick the winners before planting. The process begins with a chipper that analyzes corn seeds. The machine isolates a kernel, then uses a computer vision algorithm to orient it.
And if you kneel down and look over there, you can see the seed being presented to the blade.
The device cuts a tiny chip from the kernel while preserving the seed's viability. Monsanto now analyzes the chips for some of the 3,000 DNA markers it's identified in corn. The markers are signposts for DNA tied to traits that breeders want.
This is where the rubber really meets the road for the molecular breeding process. We're actually applying the chemistry that's going to detect the markers on the DNA.
With markers in hand, Monsanto can carry out molecular breeding, also called marker-assisted selective breeding. Here's how it works. Suppose you had two parents, each with 10 different markers linked to coveted traits, and you wanted to breed corn based on the values of these 20 markers.
To get those 20 different regions into a single plant, we need to do about 1 trillion different crosses. To put it in some perspective, there's about 1 trillion corn plants planted in the Corn Belt this year. You'd need to walk from Nebraska to Ohio and know what that plant looked like to find one plant that had the right combination of those 20 genes. We're actually able to drive those odds from 1 in 1 trillion to about 1 in 5.
Monsanto selects kernels based on the constellation of markers tied to networks that underlie yield. It then ships the chosen seeds back to the breeders, who grow the plants and mate different pairs together. From this next generation, breeders pick the best plants and send their seeds back to Monsanto for another round. Using markers and chipping, breeders can save years in zeroing in on the traits they want.
In the old biology in the plant space, it was all about what individual genes could we tweak to increase yield or have more favorable traits. So with crop yield, applying the new biology, companies and academic labs are already way ahead of the curve from the standpoint of where we're at in human biology.
U.S. corn yield per acre would grow like this to 2030 with better use of old methods. But with new techniques, such as molecular breeding, it can nearly double yield in the next 2 decades.
Science and technology provides a cornerstone that can build food security, but it can only be done if we have the physical infrastructure, the right policies in place, and the political leadership to make that happen.
It's not just food production, but virtually every aspect of human health that is being revolutionized by the new biology.
Scientists are beginning to understand that there's a window onto the world that we're suddenly seeing.
What's changed are the technologies that enable us to take those holistic views.
I think that we are, for the first time, starting to get data at the scale and of a quality that makes the analytical techniques both necessary and useful.
We are using the predictions that come from these computational systems, not as the gold standards of our biology work, but really as an accelerated roadmap to do experiments that are much more likely to be successful.
It almost seems like cheating now to be able to just look inside.
There's nothing better than watching something, watching a cell to understand why it's doing what it is, or how it's doing what it is, or in fact that it's doing something different from what you expected.
If we study any organisms, we have to sequence the genome to understand the genes.
We strongly believe the personal genomics and the personal healthcare will come. When and how, it really depends on the sequencing technology and the policy makers in the country.
Good science will not be adopted unless we can show systems that it'll work and that it's affordable.
Anything's possible. People's lives can change. Technological improvements combined with infrastructure and policy can transform households, can transform villages, can transform whole economies.
The new biology is harnessing the wave of new technologies to create unprecedented predictive models of living systems. Let's now ask the smart questions and transform the data that emerges into wisdom that can change the world.